The Interested Reader podcast is about exploring the world beyond the textbook. We'll cover many lessons in math, physics, and astronomy, and then delve into the mysteries of the universe. Physics is fun, people just don't know it, and I want to change that. I want to spark your curiosity about the world around you and ignite your passion for exploring and questioning. I'm your host, Sarah Tyke. Come with me and let's take a random walk into the world reserved for the interested reader. Hi everyone, this is Sarah with the Interested Reader podcast. Welcome to today's episode. On today's lesson, we are going to be learning about motion in a straight line in one dimension. So we are going to be starting with the x-axis motion. That's our horizontal motion. I'm always going to have the x-axis horizontal and the y-axis vertical. It's just a convention. You can always choose whichever way you want as long as your signs are consistent. Alright, so 1D motion. So we're going to kind of go through a lot of derivations of equations today, so if you're listening, this might get a little hard to listen to. I'm going to try to talk through it so you can actually at least visualize kind of what I'm talking about with the equations, but it might be a little difficult to just listen to, so I apologize for that, but that's going to be a lot of what these lessons end up doing. So from last time we talked about velocity and acceleration. And to make a little bit of a correction in notation, we talked about velocity being a change in x over a change in y. We actually want to call that an average velocity. And I'm going to denote that with a bar over the v. So v with a line over the top of it. Sometimes we'll say v bar, but honestly, after today, you'll probably never actually hear me talk about average velocity. We're going to use this so we can derive some other equations. So we have our average velocity, and that is our change in x divided by change in t. And then same thing with acceleration. A with a bar over the top is equal to a change in velocity divided by change in time. So to get the instantaneous velocity, that's where we don't have that v, or the bar over the top of it. So that's just instantaneous v. The actual notation is going to be a limit as t goes to 0. Let me just check on something real quick before I write that down. And when we're going to talk about all these motion problems, we're going to be talking about constant acceleration, so we'll get to that in a minute. Alright, so we have instantaneous velocity is actually a limit as delta t goes to 0 of our delta x divided by delta t. So that's the technical notation for velocity. It's a limiting case. So what that means is that you take this delta t in the denominator of our average velocity, and as that approaches 0, you get this limiting case of velocity. That would be your instantaneous. There's not going to be a spread of the velocity. So another way to write our average velocity is to go back up here to this delta x, delta t. And remember, we can write that as our final position x minus our initial position x naught, which is x sub 0. So notation-wise, it'll be x sub 0. I'll always say x naught, v naught, t naught, y naught. See, physics is fun. And then delta t, final time t minus initial time t naught. So again, our instantaneous velocity is this limit as delta t goes to 0. And that's also how then we end up with this quantity, this derivative, which we called last time dx over dt. So velocity is the derivative of your position. So we'll remember that here in a second. So we're going to assume that acceleration is constant. So what that's going to mean is that our acceleration, or average acceleration, is equal to our instantaneous acceleration. Okay, so if we assume then, so we have our a bar is equal to a. Let me do one more notation here. So back in our equation for average acceleration, our delta v can be written as our final position v minus initial position or velocity v naught divided by t minus t naught. So then when we assume that a is constant and that a is then equal to a with the bar over the top, we can now write our acceleration as a equals v minus v naught divided by t minus t naught. Now, in pretty much every case, we're always going to assume, or we're going to let t naught equal 0. So that means that your starting time is 0, because most of the time when you're talking about timing anything, your initial start time is 0. You can change it, you can have it, you can start at 5 minutes or whatever you want to do, but it just adds extra complications. Most of the time, t naught will always be 0, and you can always just set it that if you want. So we take our equation here, a equals v minus v naught divided by t minus t naught. We're going to let t naught equal 0, and then we're going to have a equals v minus v naught divided by t. And we're going to solve that equation for velocity v, for our initial, or our final velocity there. So I'm just going to kind of do this as a little sidebar over here. Again, if you're watching, this might make more sense than if you're just listening. So I'm going to multiply both sides by t, hopefully solving equations. As I go through and describe how I'm solving it, I hope I'm using terminology that you all understand. If not, make sure you let me know, and so I can clarify and go over more terminology, more definitions of what I'm talking about. So we multiply both sides by t to clear that denominator here. We're going to solve for v. So we just simply then add v naught to both sides. So we get v is equal to v naught plus at. Okay, and that's going to be one of our kind of foundational equations. I'm going to call that equation 1. Okay, so that is our first equation here. And now what we're going to do is we're going to go back to our equation for velocity up here. Our velocity is x minus x naught, t minus t naught, our average velocity. And we're going to take that equation plus this equation we just solved for v, and we're going to be solving for position x. So I'm going to erase all of this, and we'll start with those two equations. So we're going to be starting with our average velocity as x minus x naught divided by t minus t naught, and our v equals v naught plus at, and that's what I called equation 1. Alright, so in our average velocity, just like we did before with our acceleration, we're going to let t naught equal 0. So then we have average velocity equals x minus x naught divided by t. And we're going to be solving that for a quantity called x minus x naught. That's our change in position. So this right here is what we're actually going to solve for. That whole numerator is our change in position. And it's one of those things where it's just because you get familiar with problems, and rather than solve it for, say, x, it's easier to solve it for x minus x naught. It's just easier to solve for that quantity in the problems that textbooks use. Okay, so we've got our average velocity, our change in position divided by t. And when we let t naught equal 0, we have our x minus x naught divided by t, and that's our average velocity. So we can also write average velocity as 1/2 v plus v naught. So just like a regular average where you take your two speeds, your two velocities, final plus initial, you divide by 2, kind of your standard average. So if we take then this way to write average velocity, our 1/2 v plus v naught, and we're going to take our first equation here, this velocity equals v naught plus at. If we put that into our average velocity, then we can end up solving for our quantity x minus x naught, and we won't have to deal with our average velocity up here. Now, the reason we're going to do this, it's a little convoluted, because we want to get acceleration into the equation. And again, it's one of these things where it's just kind of from experience, you just kind of know that you need these certain equations, and when you're looking through physics textbooks, this is how they're going to do it. You want to have your acceleration in your equations. So we're just going to do this, and hopefully as I go it'll make sense. So we're going to take our equation for velocity and we're going to substitute that into our equation for average velocity. So we're going to have v bar is 1/2 of our equation for velocity here, so that's going to be v naught plus at plus v naught. So it's 1/2 times that whole quantity. So let's just kind of work through this. So we have 1/2 2 v naught plus at. And the 1/2 will cancel out that 2, so we're going to end up with v naught plus 1/2 at. And that's our average velocity. Now, again, we don't want to be talking about average velocity. So we're going to take our average velocity and we're going to be substituting it into our equation back up here for average velocity. Okay, so this is our next equation here. I'm going to just call that 2. And we're going to be taking equation 2, average velocity, we're going to substitute it back up into our equation for average velocity that says v bar is equal to x minus x naught divided by t. And I'm going to have to erase some of this stuff to get that going. So I'm going to rewrite our equation up here, kind of have a little tally of equations up in the corner. Alright, sorry guys, talk amongst yourselves. Alright, so we're going to take our average velocity, but I'm going to write this average velocity equation in terms of x minus x naught. Also, because I don't want to deal with that denominator of t. So x minus x naught is equal to average velocity, our v bar, times t. So now we can take our equation 2 with this v bar and substitute that into that equation there. So x minus x naught is equal to, again, our average velocity was v naught plus 1/2 at, and then times t. So we're just going to distribute that t across and get v naught t plus 1/2 at squared. And that is our equation for our change in position, x minus x naught. That is our another key equation that we're going to be using. Once we have, by the end of this, we're going to have a collection of equations to describe one-dimensional motion. And there's going to be, I think, 5 total. And with those 5 equations, each of them will have one unknown quantity that you can solve for. Alright, so really all we're going to be doing at this point is we're going to be taking, I'm just going to call this equation 3. So we're going to be taking equation 1, v equals v naught plus at, and this equation for change in position, x minus x naught, equals v naught t plus 1/2 at squared. So those two equations, we're just going to rearrange them and make different substitutions for different missing variables, and we're going to derive our other 3 equations from those. Okay, so what I'm going to do from here is I'm going to erase this equation for v bar because we don't actually need that one anymore. So I'm just going to leave our other equations up in the corner. So our first principal equation, v equals v naught plus at. Our second kind of principal equation is going to be our x minus x naught equals v naught t plus 1/2 at squared. I'm still going to leave that, well, I'm going to call that equation 2 now. I don't like to change numbering like that on you guys, but it'll be easier to follow. Alright, so those are our 2 equations that we're going to start with. We're going to start rearranging them. So basically there's 5 quantities that we're going to solve for in motion problems. And what they are, so we're always going to be asking for displacement, x minus x naught, or we're going to be solving for velocity, t, acceleration, or v naught, initial velocity. So those are the 5 equations, or 5 quantities that we'll be solving for in our motion problems. And we're going to have an equation for each of those. So you can say, well, I don't know, or I'm looking for, you know, just take equation 1. I want to solve for final velocity v, but I don't know what x minus x naught is. I don't have that information. So equation 1 is your go-to, because it doesn't involve, sorry about that, there we go. It doesn't involve the quantity x minus x naught. So if you don't know it, that's great. You go to equation 1. Similarly for equation 2, if you are trying to solve for whatever, but you don't know your final velocity v, that's okay, because in equation 2 you don't need to know your final velocity. Okay. So we're going to get equations for each of those 5 quantities up in that corner. Okay, so we've got our 5 quantities that we're going to be solving for. We need an equation for each of those, and we're going to start by using these 2 equations, our equations 1 and 2. That's our v equals v naught plus at, and our x minus x naught equal to v naught t plus 1/2 at squared. So we're just going to rearrange those 2 equations and make some substitutions, and by the end we'll have a list of 5 equations that you can just use to solve for each of our 5 quantities, depending on what information you have. So first we're going to take and solve our velocity equation for t. So we're going to solve equation 1 for t. And so that is just, it's, so V equals V naught plus AT. I'll just do it like this. We're going to subtract V naught from both sides. V minus V naught is equal to AT, and I always write my equations with the unknown on the left side. Again, it's kind of convention. So T will be equal to V minus V naught divided by A. So now we're going to take this value for T, and we're going to substitute it into our equation 2 for x minus x naught. So T goes into equation 2, and we end up with x minus x naught is equal to V naught times V minus V naught over A plus 1/2 A times V minus V naught divided by A quantity squared. So now it's just a matter of rearranging this. We're going to try to get our equation down to solving for a quantity V. And actually, we'll see at the end, but because I don't want to deal with square roots, we're going to leave it as V squared. But we'll get to that in a second. So we're just going to solve this equation for x minus x naught. So we're going to have V naught V minus V naught squared over A plus 1/2 A times, and then I'm going to solve this quantity squared, V minus V naught divided by A quantity squared, gives us V squared minus 2 V naught plus V naught squared divided by A squared. And then we're just going to keep simplifying down. So V naught V minus V naught squared over A plus, so in this quantity I'm going to take and cancel out my A in the denominator, so I'm going to have 1/2 A, 1 divided by 2A, sorry, times V squared minus 2V V naught plus V naught squared. Okay, so I'm going to keep solving that, but I have to erase some of the board here, so I'm sorry about this. I don't have a bigger whiteboard. Been trying to figure out how to get that to work. My camera setup is just, it's pretty janky if you can't tell already. But I'm trying to work through that. So, but for now I got this board, we'll have to make it work. And I appreciate you guys indulging me and bearing with me while I do this setup. Alright, so continuing our solving here, we've got x minus x naught. Okay, so now I'm going to multiply both sides by 2a, so I can get rid of this 2a down here and this a. So I guess I'll have to make a little more room here. So 2a times x minus x naught. So the 2a times our first quantity here gets rid of the a, so then we have 2 times V naught V minus V naught squared. And multiplying by 2a here got rid of that coefficient there, so then we just have plus V squared minus 2V V naught plus V naught squared. So I'm just going to distribute that 2 in that first quantity. So I get 2 V naught V minus 2 V naught squared plus V squared minus 2 V naught plus V naught squared. Okay, so now we're just going to combine all our quantities, and remember we got our 2a on this side, which we'll get rid of in a second. Alright. So I'm going to make some room and clear off the second half here. So our 2 V naught we have a minus 2v v naught. So those two cancel out. So we are left with a, we've got a minus 2v naught squared and a positive v naught squared, so combine those, we end up with v squared minus v naught squared, and that is equal to our 2a times x minus x naught. Okay, so at this point we just want to write this in terms of v squared. Again, if we wanted to do it as just v, we'd have to have square roots, and no one likes square roots. And I hate them almost as much as I hate fractions. So we're just going to solve this equation for v squared, which we're just going to add v naught squared to both sides of the equation. Put our v squared on the left side. So v squared is equal to v naught squared plus 2a x minus x naught. And that is one of our 5 fundamental equations. Call that equation 3. So you can see in this equation, if you don't know the time component of in your problem, it doesn't matter, we don't need it. And again, it's just, we're solving for, obviously it's the quantity v, you have to remember that you're actually solving for v squared in this equation, so you have to take the square root of whatever answer you get at the very end. And it's just a notation, that's all it is. It's just to make it easier so we don't have to write square roots of everything. Alright, so that is our equation 3. So I'm going to add that to our bank on the side. Just going to get kind of crowded up there, so we may have to I think what I might actually do, because I don't think we're just going to be using equations 1 and 2 up there, so I think we'll just get rid of equation 3. I'll write them all down at the end. Alright, so next we're going to be solving our velocity equation for acceleration. So we're going to solve equation 1 for a. So we're starting with v equals v naught plus at. Solve that for a, so we have v minus v naught is equal to at, and a, which we already know this, right, from before. A is equal to v minus v naught divided by t. And now we are going to put that into equation 2. So we have x minus x naught is equal to v naught t plus 1/2 a, which is now v minus v naught over t times t squared. V naught t, we're going to simplify this expression with our 1/2 at squared. So when I solve equations, I had to go through every single step, and if you're more advanced, you probably wonder why I do every single step, why I can't just combine them, and I just don't like to do it. I do every single step. So in this step, I'm going to be canceling my t's over here, and I'm only going to do one thing at a time per line on the equation. And these is pretty simple. You could probably combine steps, but later on, through my physics career, I found that I had to write out every single step. If I was going to add something, it was a new line. If I subtracted something, it's a new line. I'm not doing them together. So that's just how I do it. I know it takes a little bit longer. So if you're way advanced, you're probably not listening to me anyway. Or you're skipping through the lessons if you want to get to the other part. So we have 1/2 times v minus v naught over t. Oh, I'm sorry. Wrong way. See? Look at that. Okay. 1/2 times v minus v naught times t. Alright. V naught t plus 1/2 vt minus 1/2 v naught t. So I'm just going to distribute that half coefficient through, and the t. Alright, so we have a v naught t quantity here. We have minus 1/2 v naught t here, so we'll combine those. and we get 1/2 v naught t plus 1/2 vt. So we're just going to be pulling out the common quantities there, and again I have to erase this top part here, move right over there. So we're going to have x minus x naught is equal to 1/2. I'm going to pull out the way I'm going to write it is going to be a little bit weird. V naught plus v, and then t over here. So you could write it as, if you wanted, I mean, you could write 1/2 t times v naught plus v. I'll tell you, the only reason I do it is just because that's how the physics book that I was taught from does it. It puts it as 1/2 quantity v naught plus v, and then t on the other side. It's just convention. I mean, it's just whatever this book chose to do, and that's how I learned it. So that's how it just got ingrained to me. Like, that's how I memorized the equations. You can write it obviously any way you want. I mean, 1/2 t times v naught plus v is the same thing. But I'm going to leave it as written this way: 1/2 quantity v naught plus v times t. And that is another one of our 5 equations. That would be equation 4. So in this one we don't need acceleration. So we've got our quantity x minus x naught, v naught, v, t. Don't need to worry about acceleration. All right, one more. And that is going to be solving for v naught, so that we don't have to worry about that quantity. So we're going to solve equation 1 for v naught. So again, we start with v equals v naught plus at. And we say that v naught is then equal to v minus at. And that is going to go into equation 2. So we have x minus x naught is equal to v naught t, but now v naught is going to be v minus at plus 1/2 at squared. Okay, and then again we're just going to start solving. I think, did I... v minus at, oh, oops, there's a t here. X minus x naught is equal to v minus at times t plus 1/2 at squared. So we're going to distribute this t in that first quantity. So we have vt minus at squared plus 1/2 at squared. So again, we have our minus 1 at squared. We have a positive 1/2 at squared. We're going to combine those two. And we get vt minus 1/2 at squared. And again, at this point, if you wanted to, you can take that t out. You could write it as t times v minus 1/2 at. But again, it's just a notation thing. It is easier later on when you start solving the problems to just leave it written as vt minus 1/2 at squared. Just leave the t's in there. So that is our x minus x naught. So that is our last equation. That would be equation 5. So from our 2 equations in the beginning, equations 1 and 2, we're able to get these 5 equations to deal with our 5 variables that we'll be encountering in motion problems. So I'm going to go ahead and erase this work here, and I'm just going to go and write all 5 of our equations out, in case anyone's listening. Alright, so I'm just going to actually erase these two too. We'll just write them all nice and neat. I don't know, guys, I'm obsessed. Alright, so we have, in summary, V equals V naught plus AT. We have X minus X naught is equal to V naught T plus 1/2 AT squared. We have V squared is equal to V naught squared plus 2A times X minus X naught. And then we have X minus X naught is equal to 1/2 V naught plus V times T. And X minus X naught is equal to VT minus 1/2 AT squared. So those are our 5 equations for motion in one direction. And again, this is going to be in the x direction. That's why we're talking about X minus X naught. And the caveat for all of this is that we have to assume constant acceleration. And that's kind of a standard condition in physics problems. So it's not too unusual. So these are the starting point. You can memorize all 5 of them. I think as a student we all tried to do that because it seemed easier. But if you just remember the first 2 equations, 1 and 2, you can derive everything you need from these 2 equations here. So we started with equations 1 and 2, solved them for different variables, and just basically we're solving equation 1 for a variable and putting it into equation 2 to get the other 3. So really you could just remember 2 equations and then derive what you need from there. Okay, so this is one-dimensional motion. Next time it's going to be a super easy lesson. We're going to talk about motion in the y direction. Alright guys, that's all I got for you as far as the lesson. I'm going to take a quick break and we will continue with the rest of today's topic. And I will see you guys in a couple minutes. Thanks guys! Hi guys, welcome to the Interested Reader podcast. I'm Sarah Tyke, and hopefully you were able to sit through the whole lesson earlier today about our one-dimensional motion. If not, I don't blame you. It's all good. So the topic of today's podcast is going to be, it started out as the DNA phantom effect. That's what I wanted to talk about. But in order to get there, I had to do a lot of research on what's called wave genetics. That's kind of where all of this originates. So I've read about the DNA phantom effect in this book called Source Field Investigations by David Wilcock. I love that book. And it's just kind of a couple pages, just a small section in this book about DNA phantom effect. So I thought that's pretty cool. I'm going to look into that. But it all originates from this idea of what we call wave genetics. So we're going to start talking about that, and then we'll get into what the phantom effect is. And we may extend this into another episode about wave genetics, because it's pretty cool and there's so much information that you can just really kind of get lost in all of it. So wave genetics, it starts with a Russian scientist, Dr. Peter Garyaev, and he died in 2020 just after he received a Nobel Prize nomination. So he has a website still, wavegenetics.org, and we'll look at that in a second. So he's considered to be the founder of this idea of wave genetics, where genes function as waves. And this idea was actually first proposed by another Russian scientist, Alexander Gurevich. He was a medical scientist, a biologist, and he kind of proposed this idea in the early 1900s. Didn't really have any technology to to prove it. He just kind of speculated that a wave theory might be a better way to explain genetics than the typical molecular or particle view that the Western scientists have. It's kind of interesting because when you go through and research all this, a lot of the information is from Russian scientists, so a lot of the papers are in Russian, if they got published at all, because a lot of this research was done during the Soviet era, and their scientists, they weren't publishing their information to the world, so a lot of it just never got released. And it seems to be the split between kind of the Western scientists going the route of particle physics and modeling everything as particles and molecules, and the Russian scientists seem to go and take a field theory approach. So everything is about energy and waves and fields. And so it's kind of interesting to see the difference in there. The problem is that a lot of this research then sort of gets belittled by the Western scientists because they're like, a wave theory? Like, no, I don't think so. It's a particle, clearly. It's all particles. So when you start talking about fields and waves, a lot of the mainstream science really won't hear it because they just think that it's pseudoscience. But it seems that all the Russian scientists went this direction, and they did a lot of cool stuff. And they'd have experiments that they did to show that their theories were working, at least once they got the technology. So back to Alexander Gurevich. So in 1910, he promotes this idea of morphogenic field theory. So what he said was that you could take a group of cells and that they respond to biochemical signals, and then they develop into structures or organs. And these cells will produce an energetic field that contains that genetic information of that cell. So you'd have like a group of heart cells that would create an energetic heart field. So if you put new cells in that energy field, they would become heart cells or heart tissue. And he had postulated this is what was going on when you have like salamanders that can regenerate their limbs or their tails. Those cells are producing an energy field that has the genetic information of a tail or a limb. And then when you put new cells in that area, they are using the energy, the information in that energy field to grow into a limb or a tail. And again, this is 1910, so he didn't really have any way of proving it. It was just more of a theory. And then Dr. Garyaev is kind of credited with turning that theory into a science, and he actually uses it for healing. And if we'll take a look at his website over here, you go to wavegenetics.org. You get to translate it from Russian into English, and there's I don't know if this is a chatbot or an actual person, but the window keeps popping up, so you may hear a little ding every now and then, and because I'm going to have this website up, and it's in Russian, so the customer service rep is typing in Russian or asking questions in Russian, and I do speak a little Russian, but not much. So I can tell that they're asking if I need help, but I don't know how to respond because my keyboard's not set up for Russian on this one, so I apologize if you hear a ding off and the chat window pops up. So this is Dr. Garyaev's website. So he does a thing called linguistic wave genetics, and the linguistic part is a sound component, and we'll talk about this later, but basically he's using sound waves to carry genetic information. I mean, it sounds so fantastical, but he has results. He has people that he treats with this, just a sound field, and I mean, they report healing. Obviously it's testimony. I don't know if you can call it actual scientific evidence, but if someone says that they were healed, they see results, and we'll get to all the results of this stuff. And it's amazing. Okay, so this is Dr. Garyaev's website, Linguistic Wave Genetics, and there's a pretty cool website where you can kind of go through. So here's a little bit of a biography about him. So in 1984 is when he first kind of discovers this DNA phantom effect. It doesn't get published until, I think, 91. And then again in 2020 he was awarded a Nobel Prize, but he died shortly after. Or he was nominated for a Nobel Prize. And this is just a little bit about some case studies that they've done. And again, the site is in Russian, so you have to translate it. And so if you're watching, I'm scrolling, it takes a bit to buffer into English. And somewhere on here, when you go to the page, they do have an example of one of his WAV files. But I had to close it out so I could navigate the website, and I don't know how to get it back. So I've listened to it, and it is not fun. It's not a pretty sound. And we'll get to this later because that happens. I have another story about that later, about the sound of these things. Okay, so we've got Alexander Gurevich in 1910 talking about cells producing energetic fields. And an American biologist around the same time, Ross Granville Harrison, had also discovered these fields of cells. And he found that when you put undifferentiated cells in these fields of like the limbs or the tails of salamanders, they would develop into the corresponding organ. So it seemed to support this theory of this wave genetics, where there's an energy field that contains genetic information that can tell a cell what to develop into. So we come to the 1930s and we end up with the study of genetics and the discovery of chromosomes. And that's when it kind of minimizes this idea of field theory, because now instead of having an energy field that you can't see, now we have chromosomes that are tangible, physical objects. So that's kind of where, again, the Western scientists kind of latched onto that and said field theory is nonsense. The chromosomes, where it's at. It's all particles, it's all physical stuff. Which, I mean, it makes sense, obviously. But they completely dismiss field theory like it has no scientific backing or scientific merit at all. And it does, and the Russian scientists went that direction and have found that field theory actually seems to be quite valid. So, for a long time, field theory is kind of minimized, kind of put on the back burner. But in the late 20th century, researchers kind of come back again to this idea of field theory and field concept. And again, we have this, Dr. Garyaev gets in and starts studying this. And one thing that I'm going to call them mainstream scientists that were using chromosome theory, they discovered that different genetic mutations cause the same malformation. So you have a genetic mutation, but it can cause two different deformations or malformations, and they couldn't explain why that is. You should be able to just say, this defect causes this malformation, but that wasn't happening. So another American biologist, Scott Gilbert, suggested that genes are acting on a field, an energy field, and that field then acts on the developing organism. So you could have a mutation creating a sort of field, and that field acts on the organism, and it could change the malformation depending on the organism that's in that field. And all of these studies where we're talking about growing cells and putting cells in fields, all this takes place at an embryonic level. So that's when you have your cell differentiation, like your stem cells. You're not just taking an adult, cut off their finger, and then putting their finger in a field and growing a new arm or a new hand or something. It's all taking place at the embryonic stage when you have this rapid cell division and differentiation. So Gurevich also discovered what's called a biophoton. So he discovered that photons are emitted by biological tissue. And this, you can see them, they're weak electromagnetic waves in the UV spectrum. And so he believed that this photon emission allowed his theory of morphogenic fields to be real, to be realized, and to influence cell development in embryos. So, and again, I'm not sure exactly how I don't know how he actually found or discovered the photons being emitted, because it wasn't until 1962 when they were able to really confirm that this does happen. And maybe Gurevich just had some equipment but just not good enough, where he, it wasn't consistent results. I'm not really sure. But in 1962, once they got some more technology where you have a photon counter multiplier, then they were able to really determine that, yes, biological tissue, biological cells do emit photons. Because at this point, it's hard to detect just one photon. So now we have technology where you can multiply the photons, so you can really see the effect, and you can detect them down to much more sensitive levels. I think they can actually detect down to single photons now. So in 1910 they couldn't, when Garyaev was proposing this. So in 1962 they confirmed biophotons. This is a real thing. And then in 1974 experiments are duplicated, and back in 1974 the Russian scientists claimed to have detected intercellular communication via these biophotons. So cells are using these photons, these light particles, to communicate with each other. And again, all that was in 1974, and the experiments have been repeated in labs all over the world. So biophotons that Garyaev proposed in 1910 have been proven to be a thing. They are real. And I haven't found the paper on this, or where the actual proof of this, but I have heard multiple times that at conception, when the sperm and the egg meet, there's actually a flash of light. And I'm assuming they just detect this all, in vitro. But there actually is a detectable flash of light at conception, which I think is pretty cool. So that means when a new person's being created, light is being produced. And I love that. I think that's so cool. So Garyaev's work also influenced Rupert Sheldrake, which, if you remember, we talked briefly about in our last episode when we were talking about scalar energy. So Sheldrake had come up with this idea of morphic resonance, where you have this field of information connecting all your life forms, which would probably be the scalar waves. That was kind of the topic of our last discussion, where scalar waves are carrying information, instantaneously. It's kind of how you can feel that somebody's looking at you. You don't see them, you just feel it. So there's something being communicated between two people where you kind of feel that they're around. You can feel that they're looking for you. And I think that staring effect is something that Sheldrake also came up with or studied in detail. So Sheldrake was influenced by Garyaev's work. All right, so that's a little bit about our wave genetics, and just this idea that genetics, that our DNA is actually energy, that that is a wave form, and that it's influencing some field around it. The point of this book, this Source Field Investigations by David Wilcock, was that there is a source field that we are all living in, and basically energy imprints on this field, or this field can carry information. Again, we get into the scalar wave theory again. So when you have an energy field, it basically makes an imprint in this source field, or a scalar field that we're all living in right now. And then the information just stays in that field, and then it can be used to convey information, whether it's genetic information, whether it's a thought, feeling, who knows what else. So that's kind of where we're at with this, with our wave genetics. That's kind of the background here. And also, as far as mainstream science goes, you probably want to look into the Higgs field, because I believe that is kind of what mainstream science would call, what David Wilcock calls the source field. What we've been talking about is a scalar field that we're all living in. Ancients called it the ether. Modern science kind of calls it the Higgs field, with the Higgs boson. That was a big thing. So, that's our background. We're all in an energy field, and DNA, genetics, basically any physical living object has a field around it that influences this field that we're in and conveys information. All right, so that's kind of like our morphogenic field theory, our wave genetics. And again, if you look at this website from Dr. Garyaev, there's a lot of stuff here. You can click on, they've got like a theory page where they go into more about what they do quantum consciousness, essential quantum dualism of the gene. So that's why this, and there's a lot here, and so I might go into a little more detail of this for another episode, but wave genetic code. And there's our little chatbot again. Sorry, guys. And sorry, chatbot. I'm just hanging out. So Dr. Garyaev has done, studies with actually healing people. And again, we'll get to that a little bit later. We can look at some of the examples. But so his website has a lot of information on it. Here's the matrix, the sound matrix. I'll leave this up and we'll come back to this. Because again, part of his theory is that you can heal people with a sound file. And we'll get into that and then we can come back and listen to what it sounds like. 'Cause like I said, it's not, it's not pleasant to listen to, but there's a reason for it. Okay, so that's our background. So now let's get into what I really want to talk about was the DNA phantom effect. That's what kind of started all this. So this was first discovered by Dr. Garyaev in 1984, and he didn't publish it until 91. So the idea was he takes a DNA sample in a test tube, shines a laser light through it, and then you collect the light that scatters from that DNA molecule. So okay, no big deal, you collect your sample, you can, by the way the light scatters and how much you can collect, you can see, okay, there's a DNA molecule in my test tube. So then what he did was that he took the DNA sample out, took the test tube away, everything, and he found that there was still light being scattered. He was still collecting photons. He was still collecting energy from the place where the DNA sample was. It wasn't there anymore, but he was still collecting energy as if it was. So that was a huge surprise, because once you take the sample away from the light source, there shouldn't be any scattering. The light should just go straight ahead. There should be no scattering. But that's not what they found. They found that when they take the sample away, light is still scattered as if your DNA molecule was still there. So if we just, on this website here, I can link all these in the show notes because they're kind of long websites. But this is kind of a picture of their setup here. And the black and white photo, if you're watching, is, I can't really tell much of what they're doing, but so here they have below it is a diagram. So they have a laser that goes through a collimator, which just makes sure that the light is a column, that it's not scattering. And then you've got your little test tube here, your cuvette with your sample. It scatters the light into your detector, your multiplier. And then you come out to the your computer screen, and you can read how many photons you're detecting and what pattern. So on the screen here, again, if you're watching, this is the diagram of their setup. So then what they found, here are the results. So the background is just empty space. You kind of figure that, yeah, with no sample there, you're going to have some background. There's going to be some scattering because there's going to be dust and particles in the air. But it's pretty random, pretty low count. Your photons per channel go from like 624 to 784. So not even a range of 100 photons. So basically nothing there. So here they put their physical DNA in a solution. And you basically, this was kind of weird too, so you get this sinusoidal, and maybe this wasn't weird. You get this kind of sinusoidal pattern that sort of decreases in intensity over time. But clearly you can see that there's a physical sample there. There's something scattering the light, and the intensity goes, in our background it was a couple hundred, 600, 700. Here it goes up to about 60,000 photons per channel. So clearly something is in the way of that laser scattering the light into your detector. It has this really cool, I guess, a sinusoidal pattern. So then they take the sample out. Again, you should go back to this background empty space, no sample, should be no scattering. But instead they find this picture here. Phantom DNA. So there's just these large peaks of photons being detected. It's not the same pattern as when the sample was there, but you have peaks that go up to about 600 photons per channel, and then it drops down to about 0. And then there's another peak over the total time here is about 3 seconds. And these are channel numbers, so I'm not exactly sure what that means, but if each channel is picking up about 600 photons. So there's definitely something registering, because again, it drops down to like 0, and then it pops back up, and then drops to 0. So there's something there. And it's a pattern too. Again, it's not like that random scattering that we saw up in the background where there's no sample or empty space. And then after about 25, 26 seconds, you have another pattern here where it's basically all 0, and then you have these 3 large peaks that go to about 30,000 photons. So again, there's something going on there. It's not 0, it's not your same background pattern as if there was no sample there. There's still something being detected, even after the DNA is gone. And not only was that enough of a surprise, the fact that you're still registering energy after your sample is gone, that was a surprise enough. But sometimes you could detect these photons up to 30 days after the experiment. I mean, 30 days and you still have something in that field. There's still something emitting photons, even though the physical sample is gone. So that suggested that the DNA is somehow making an imprint in this field, this energy field, a source field, a Higgs field, scalar field. I don't know what you want to call it. But somehow the DNA is making this energy imprint that just persists even after you take the sample away. Now, is the scalar fields, because we just talked about how scalar fields can carry information. So maybe we're in a scalar field and they are actually holding the information of that DNA that is actually a wave. Because with most of quantum physics now, I mean, I think for a long time we've all understood that you can represent things as a particle or a wave, and that's how we're taught in school. So down to the very smallest molecule, why not look at everything as a wave? I mean, if a proton can be a wave and a neutron and an electron, they're all waves, and those make up the bases that make up our DNA. Those are all waves. Everything can be a wave. It's interacting with this scalar field that we're in, and it just makes an imprint in it. So that's really cool. So this is the data that was collected just on a graph, which is pretty cool. But if we go over to this paper here, so this is, let me just scroll back up to the top. I had the cool part. So it's a paper called Crisis in Life Sciences, The Wave Genetics Response by Dr. Garyaev, Friedman, and Lenova Garyaeva. So this was their paper where they published their findings about the phantom effect. And so they're going to go through a little bit about genetics, the kind of the traditional genetics here. But so here we get to their phantom effect, and so they're going to set it up. And I'm just going to read a little bit from here. So it says that, so they find this phantom registered using laser spectroscopy. So you find this phantom effect, and you spray gaseous nitrogen in the area to it's supposed to clear out the area, it's supposed to disperse the energy. That phantom effect comes back in 5 to 8 minutes. The phantom disappears completely after 1 month. We remark that sound waves radiated by the DNA molecules were registered in these experiments. So the authors found that they could pick up sound waves from the DNA molecules. And that is where Dr. Garyaev uses this idea of sound waves to heal people, which you can find on his website, this linguistic wave theory. So the DNA is emitting a sound wave, and that sound wave is then essentially imprinting in this field. So here again in this paper, this is where they're going to publish those graphs that we just looked at with the background space, the physical DNA looks like the sinusoid, and then you got the two phantom graphs. So then here again, I'm sorry you guys got to be watching this, but or you can search for this paper. I can put a link in the show notes. But so here's a picture, like an actual visual picture of what they're seeing. So if you're watching on the screen, on the left side, this is their setup where they don't have any sort of electromagnetic field turned on, like their lights, because the idea is they have to shine a laser through the DNA sample. So on the left here is their setup. Nothing's turned on, it's just sitting there. Right in the center is where they have their test tube, their cuvette, with their DNA sample. So nothing's really going on there. And then they turn on their laser light, and on the right side, you see these white shapes off to the right of their sample. And nothing's moving, because the picture looks like if you took and you moved something, you kind of get that ghosting effect, or like, you know, if your cursor's on your screen and you move it too fast, you kind of see the trail of the cursor, that kind of ghosting effect. That's what it looks like in this picture. But nothing is moving. Their system is completely stationary. All they've done between before and after is turned on the laser. So they're shining a light through the DNA sample. And somehow, apparently, that light seems to energize the DNA, and it produces this field next to it. And it's not just the DNA. You can see it's their equipment, too. There's like a little, I don't know, a tube-looking light that they have. Like if you had like a incandescent light bulb as a tube or something. And you can see that sort of image ghosting off to the side as well. And then in the DNA sample, it's hard to tell, but you can kind of see that there are almost like pulses to it. So you can see like there's a brighter part in the image, and then it fades, and then bright and fades, and bright and fades, kind of like a wave. Like you would expect when you have that sinusoidal curve, the ups and downs. So that's pretty cool. I mean, you can definitely see something is there. And again, it looks like if you were to move your equipment, but they're not moving it. They just turned on a light. And you can see these images. So I'm just going to read from here. So it says that the amazing phantom structures were found to move along complicated trajectories. Moreover, they mimicked the shape of the DNA sample and some objects around it. So we're seeing this phantom effect. Not only is it there, but according to the authors here, it mimics the shape of the DNA sample. So it's actually that spiral helix DNA. So that's their image from this paper. And again, they have the physical objects too, the non-biological samples also have that same phantom effect. So we'll just scroll down here for a second, and we'll get to this one in just a minute. The phantom leaf effect is coming up. So this was just a visual picture that they actually did capture this phantom on film, which I thought was pretty amazing. So let me just... Okay, so this was our visual. The next thing that they did, which was pretty cool, was this PCR amplification of the phantom DNA. So let me click over onto... Oh, here's another picture of some different stuff some different images. So this is from another paper by Garyaev. So here is their original photo that we were just looking at with their light set up. They turn it on, and then you can see these ghosting effects off to the side. So here's some other pictures. Honestly, I can't tell in these pictures what they're trying to show, and we have to look at them backwards. So the before picture is actually on the right. In this image that I'm looking at here, it's like looks like a lampstand with some red twisties. I don't know if it's a cable or a wire. It just says that to the left is the original image, and to the right is after they've turned on their sampler, they're measuring. And they point out in the photo on the right where the wave the phantom is, but I don't see any difference between that and the first one. So maybe you want to take a look at it, and again, I'll post the links in the show notes, but I couldn't find any difference. It looks brighter because the light's on in the after picture, but I don't see this wave replication that they're pointing out. I just don't see it. And then they go into some different, you've got red, blue, green filters. Again, I don't see any difference. Here's another one. It looks kind of like a different angle maybe of that one that we just saw with looks like the lamp with the red wires or filaments. Again, the before picture's on the right and the after picture's on the left. And they're pointing out the phantom wave replication. It just doesn't look any different to me than the first one. And I don't know what that red filament is. It almost looks like a ribbon cable like you have for a printer or something. I think you use this for printers, maybe monitors. But those flat ribbon cables. It's kind of what it looks like what is in the picture, but I don't see any difference between before and after in these. But again, maybe you guys take a look at it and maybe you can see something I don't. But so those were just some visual data of their phantom effect. This one, they're kind of doing something different where, it's the same setup as in our first picture. We've got your laser light, and when they disturb the setup, so like somebody goes and pokes the sample where the DNA is to kind of jostle it a little bit, the phantom image disappears, but then it also shifts over to the other side of the setup. So it was on the right side, you disturb the camera, you move it a little bit, or the sample. The phantom disappears, and then it reappears on the left side. I'm not sure if they ever found a way to figure out why that happens, just that it did. So here, there's a picture. It says disappearance of DNA sample, of DNA wave replica after 5 to 8 seconds following the interference with the DNA sample. So somehow when you mess around with the system while it's on, that phantom effect goes away. And so here's just more, like I said, the pictures to me look the same. They're a little bit colored differently because the light comes on, but this paper is pretty cool as far as if you want to look at some visual representation. Again, maybe you guys can see something in the pictures that I don't. They just go on to their methods here, the setup of what they did. So this was pretty cool. So this was your visual representation of the phantom effect. And I mean, definitely in the first picture you do see something off to the side of the setup once they turn that light on. There's definitely something there. Okay, so that was the visible images. So we want to talk about PCR of phantom DNA. So if you don't know what PCR is, it's polymerase chain reaction. Probably by now everybody knows about it because those were the tests that were being used during COVID to determine if someone had COVID, and they're not diagnostic tests in any way, shape, or form. I was horrified that they were using PCR tests for diagnostics because they're not diagnostic tools. We used them in the lab where I work all the time. And the whole idea is to amplify a sample. So you take a few copies of your sample, whatever it is. In the lab where I was working, we had genes. We were making genes. And so you only had a few copies of them. The idea is you put it in this PCR, thermal cycler, and you make tons of copies. You amplify it. The idea is just make more and more and more of the stuff. So for diagnostics, it's not good at all because you are essentially artificially creating something that isn't really there. You're making more of it. So anyway, so you take PCR, polymerase chain reaction. The whole idea is to amplify something that isn't there, or not that isn't there, but if you have a small quantity, maybe you have one copy of a gene and you want to make more. You run it through PCR a few times and you can make more and more and more, millions of copies from just a single one. So that's the whole point of PCR. So what they're going to do is they're going to basically take this phantom DNA and we're going to make more of it. So what's on the screen right here is just a typical, what we call as a gel. So when you're doing PCR, you have a gel. It's just consistency basically of Jell-O, if you've never worked with gels before. It's an auger. You warm it up, you pour it into like a little plastic box, and you have what you put at the top of it, you put what's called a comb. It gives you, oh, sorry guys. I think if you've seen all that ABG stuff just popped up. So in this picture here, you can see all these green little bars across the bottom. So that's kind of your starting point. So you put a comb in this gel, it solidifies, and then you take the comb out, and now there are these little wells in your gel that you put your DNA sample into. And your sample is going to have dye in it, so it fluoresces like this green right here. And then take a picture of it with, we used to use computers, but you can make actual physical, like film prints of it. So what you're going to be doing with PCRs, or with a gel, you put your sample in the well, and it's basically going to go from the top of the gel down to the bottom. It's going to work its way down because you're applying electricity to the gel, and our DNA is polarized, so it has a positive and negative. So the DNA works its way through the gel, and you'll see these what they call bands. So in this picture, you see these green bands, and those are just different lengths of the DNA. So when you see something that's really bright, like this one right here, the really brightest green, you know that means there's something there. There's some of your DNA sample has made it down to this part of the gel. So that's how you do the gel. I think I misspoke when I said that this was PCR. This is how you run a gel. This is what we're looking at. And then that's usually how you tell if you have sample or not, is you run this gel, because if there wasn't any sample, like you don't have any DNA in your test sample, you wouldn't see any of these bands like over here off to the side. There would just be blanks. So in the lab where I was working, we would do this, and it was heartbreaking because you'd spend 3 days working on building up this gene. You go and you run a gel, and you find out there's nothing there. It's completely blank. So you're like, what happened? That was like 3 days of work. Nothing there. So what you're hoping to see are these distinct bands like that, and then you're like, yes, I got sample, you know where it is. Okay, so that's just a little bit of background of what we're looking at here as far as a gel and what PCR is. PCR is going to be, you take your sample, you add in some other components. They're called primers, nucleotides, the A, T, C, G. You're going to add all that into a mix, and they're going to do it's called thermal cycling. So you're going to heat it up, cool it down. Heat it up, cool it down. You're going to keep doing that, and you're just going to make more and more of these things, of whatever your target copy is. All right, so this just goes through a little bit of their methodology. They give the sequence that they use. They give their solution, their whole, whatever they use. So if you had the equipment, you could replicate this. Oops, that's not what I wanted to do, I guess. Okay. So this is just sort of just their methods for it. I don't see their amplification. This was their method, but I don't see their findings. Well, okay. Let me just... Well, anyway, so what ends up happening here, since I can't find the actual file, there were so many that I had, I must have linked to the wrong one. But, so what happens is they take a DNA sample, you shine a laser on it, and then you take the resulting electromagnetic radiation and you record it as an audio file. So if you remember, I just talked about how in that last paper, Garyaev had said that the DNA molecule, they recorded a sound file from the DNA molecule when they were doing this phantom effect. So you shine the laser on your DNA, they say for 3 minutes. There's the sound file that they record. It says recorded by a radio at 700 kilohertz, and then it was converted to a sound file, an audio file. So then they took this audio file, they played it next to a test tube that had purified water and no DNA. Okay, so you've got your DNA sample that has your laser on it. It's resonating. It's producing a sound file or a sound that you record. Turn it into an audio file. You play that audio file next to a tube that has just purified water in it. No DNA sample. You do PCR on that test tube. And remember how I said that PCR will just take like one copy of something and amplify it, because you're using PCR to detect, to make more of something when you have a very, very small amount. So they do PCR on this purified tube that had no DNA in it. They run their gel, and they found that there was DNA in the test tube. So, think about that. They had a tube that had no DNA in it, purified water. They play a sound file that is supposedly the sound of DNA. And then they find that there was DNA in the test tube when they do their PCR. Where'd it come from? You play sound and you have just now created DNA, or you've changed something in that water. So the theory that Garyaev and his colleagues postulated was that sound can carry torsion information, including information about DNA, which is their hypothesis. So torsion information is that's basically where you have a spiral, like a vortex, which actually comes into play a lot in a lot of the weird sort of sci-fi, not really fiction. You talk about like torsion weapons, and torsion physics is a big thing when you get into like time travel and bending space and time. And so DNA is it's a spiral, it's a helix, and it's rotating. So their hypothesis is that sound can carry torsion information, including information about DNA. That is pretty cool. Let me see if I have this link. Maybe I just need a different link here. I don't know if this will work. So in one of the papers, you can actually see their results of their PCR. Because from what I have on the screen here, I don't see their results. I see that this is their positive strand where they're going through what they did. I think, this is the one where they have a link to download the audio recording of the DNA that they used in this sample, but when I tried it, it just took me to like a cloud storage site that I guess you probably have to sign up to get their file or whatever, so I didn't do it. I think, I think if I look at this one here, so it was a little bit difficult to look at in this picture, so I'm sorry guys, I'm just kind of scrolling through trying to see if I can find out where the right picture is. I think this one was more just instructions on how to do it. I don't see the link for their thing, for the actual results. I did find it in some paper, but I just don't have the link for it on me at the time. So, but the idea was that I mean, you really did see, like this picture here that I'm showing of their DNA with the bands. After they ran PCR on a tube that should have just had purified water in it. There shouldn't have been anything in there. You should have run your gel and had nothing show up. But you definitely see DNA in that sample. After playing a sound file next to the test tube. I mean, how cool is that? That is just amazing. Okay, so that's the DNA phantom effect. So we have basically this idea that DNA is somehow affecting the atmosphere around it, if you want to call it atmosphere. You can call it a field. But you can detect photons where the DNA was, and according to Garyaev, it maintains the shape of the DNA molecule up to 30 days after you take the sample out of the room. So what is going on there? Somehow the DNA is still emitting light, it's still emitting energy even after it's gone. So it's imprinting in some sort of field that you can still detect up to 30 days later. So that was the phantom DNA effect. So next I want to just talk about the phantom leaf effect, because it's much more physical, much more visual. And this was noticed first by Russian scientist Adamenko, and he discovered that if you cut off part of a leaf and you put the rest of the leaf in a high frequency electromagnetic field and you take a picture, you'll see the whole leaf as basically an energy field. So you'll see this energetic remnant of the part that you cut off. So he's basically discovering that everything has an energy field. And the technique is known as Kirlian photography. So that's Kirlian, named after Semyon Kirlian, a Russian scientist who apparently accidentally discovered this phenomenon in 1939. So what he was finding is, I'm not even sure how you accidentally do this, but he has an object in a high magnetic field or electromagnetic field, and he's taking a photograph of it, and on the photographic plate he sees this energy field around objects. And again, it's one of these things where you'd probably hear this as an aura if you're talking about, either alternative healing or people that work in energy healing like Reiki, things like acupuncture. You may hear people talk about an aura. A lot of times with paranormal shows, a lot of times they'll talk about people having an aura around them. You can see it like on the IR camera, infrared camera. But this wasn't even that. This was just, I'm not sure, I think it was just a visual, I don't know what spectrum of field they use. But so the idea is that if we have this energy field around us, this aura, alternative medicine will use it to detect disease, because the theory is that if you're sick, your energy field won't look quite right. It should be like kind of complete and vibrant, and I don't know if I want to say like it extends very far from you, but it should at least be kind of complete around your whole body. And if you're sick, you might see gaps or holes in this energy field. And then alternative medicine would use that to say, well, you might have a sore throat, or you might have this or that, because your energy field is not complete, or it's just off, however you would calibrate it, I don't know. So Kirlian photography. So you can search for tons of this stuff online. There's tons of pictures. So here's just a few here, if you just search for phantom leaf effect. So this picture here with this kind of yellow leaf right here is one of the good ones. So this, you can see that the yellow part is where you still have a physical leaf, and they have a mark on here that says cut line. So they've cut off the tip of the leaf, and then they put the leaf on this photographic plate, and they take a picture, and you can see that where the leaf has been cut off, you still see the outline of that tip of the leaf. I mean, it's a perfect outline. And you can see all around the rest of the leaf where it's still the whole living leaf, you can see like a white aura. around it. And that aura just goes all the way around, even where you cut it off. Like it's still there. And then you can show a lot of these other pictures, the same thing's one over here on the right or on the left, just black and white photo. So they've cut off about half of the leaf, the top half. And the physical leaf is down here, you can see it's like kind of bright white. And then the top half, the tip of it, still has an energy outline, like perfect, like it's still there, like it wasn't even gone. But it is. So there's still some sort of energy that's keeping the shape of the leaf when you take a picture of it. So this was all the phantom leaf effect. Just a couple more like enlarged images. So here's that black and white picture here with the leaf, where the top half's been cut off, but you still see the phantom region. I mean, almost perfectly. It's almost as, the outline is definitely as solid as the rest of the physical leaf, but there's nothing there. There's physically nothing there in that top region of the leaf, but it shows up on camera. So that, I mean, that's pretty cool. So Kirlian photography, if you search for it, you'll get tons of results, even on how to do it. So here's just a Pinterest page of just a bunch of different examples. So here they've got broccoli. You can see there's a little purple outline or aura around it, butterfly. So this picture here is of two people, and you can kind of see an energy field around them, and that's where you get this idea that people have auras, and where you may have like psychics or whatever talking about how they can sense an aura. But yes, you can find, here's an onion. They do a lot of, I mean, even inanimate objects. I mean, there's wrenches, coins. All these things are, when you apply this high magnetic field, electromagnetic field to them, and you have a photographic plate. Again, there's a whole setup. There's instructions all over the place. I do know that it requires high voltage, so it's not recommended to just kind of carelessly do this. You do have to kind of, you know, research it, look into it, make sure you got all your safety precautions, because you are dealing with high voltage electricity. But you can do these. You can make them, you can take pictures like this yourself where you can see the energy, the aura around objects. So that's what Semyon Kirlian discovered. And so he did the phantom leaf effect, which just seemed to kind of support this idea of field theory, and that physical objects have an energy field, and we're influencing or imprinting a field around us, and that imprint stays there even after the physical object is gone. Like when you cut the leaf in half, you will still see the energy outline of that leaf. Okay, so I'm going to go, and we're going to go back to this paper here, this Crisis in Life Sciences by Dr. Garyaev. Here he talks about the phantom leaf effect. Just a little more of a diagram of what they did. So there's this thing called a cytopathic mirror effect, or a pathological mirror effect. So basically what they do is they take two cell cultures, they're in sealed glass containers, you put a quartz plate between them, so they're two identical cultures, and then you introduce some sort of pathology into one of the cultures, whatever that disease is that you want to introduce. You introduce it to one culture, and within 2 to 3 days, the other culture that is completely separated, different container, sealed glass container, separated by a quartz plate, that second culture will exhibit that same pathology in 2 to 3 days. And it's not anywhere near, it's sealed, completely sealed off from the diseased culture. But somehow that pathology is being transmitted to the healthy culture, and that healthy culture will exhibit the pathology in 2 to 3 days. So it's called the mirror effect. Transmission of morphogenic information. So this is where they took a laser and they shined it through salamander embryos onto frog embryos. And the frog embryos then turned into salamanders. So somehow this laser light goes through a salamander embryo, And then that laser light goes into a frog embryo, which should come out to be a frog. But then those frog cells, those frog embryos, turn into salamanders. So somehow that laser seems to have picked up the DNA code from those salamander embryos, transmitted to the frog embryos, and the frogs developed into salamanders. It seems, like science fiction. So that's morphogenic information. Similar thing happened with raspberries, where you take a laser light, you shine it through a healthy raspberry tissue. So the laser goes through a healthy raspberry tissue onto a tumorous cell from a raspberry plant. And then several months later, that tumor develops into a healthy raspberry plant. So you've taken a healthy cell, laser goes through it, laser seems to pick up that genetic code of that healthy raspberry cell, laser goes into the tumor cell becomes a healthy plant. So it's like you're conveying health information with a laser. And we'll come back to this here with Dr. Garyaev's site. And here's something that was really, I think, pretty phenomenal. It was wave genetics healing diabetes in rats. So on the screen I just have his paper on here. I'm kind of going through this. So they took 2 groups of rat. The control group had a lethal dose of poison administered that destroyed the pancreas. So they developed type 1 diabetes and die within 4 to 6 days. That was your control group. You don't have a pancreas, you're going to have diabetes, you're going to die. So the experimental group was given the same dose of that poison, but at a critical condition, right where they were kind of at the point of no return, like right at that cusp where they're, you're about to die, basically. At a critical condition, they were treated with, quote, a healing wave information. After they had this healing wave, this sound wave applied, we kind of talked about how the DNA had a sound to it that they recorded. Same sort of thing here. They recorded a healing wave. They play it to the rats. I don't know, I think we'll get to how they did that maybe. Or they just played it around the rats. And their blood sugar normalized. The destroyed parts of the pancreas were reconstructed, and 90% of the rats lived. Now remember, they had a poison that was administered that was supposed to destroy the pancreas. And it wasn't like they were healed like seconds later after giving them the poison. They were allowed to progress to the point where they were critical, almost going to die. They have this healing wave information applied. The pancreas reconstructs, and 90% of those rats lived. So this healing wave was produced by, quote, a biocomputer when the laser beam scanned the healing matrix, unquote. And the healing matrix was the healthy pancreatic cells from the newborn rats. So they take healthy pancreatic cells, shine their laser through it. They record, because we talked about how DNA has a sound associated with it. So they record that sound file as the laser beam goes through the healthy cells. Record that sound. That becomes your healing wave information. You play that in front of your diseased rats, and somehow that sound wave conveys the healthy information of that healthy pancreatic cell to the diseased cell, and the cells heal and regenerate. So the information for the healthy pancreas was stored in the DNA of the healthy rats. And then this information was passed to the sick rats and programmed their stem cells to regenerate their pancreas. Isn't that insane? So you can just go through and you can read this paper. They've got all the data of, what they did, and their methods. So the last part of this paper that I'm showing is a new paradigm for life sciences. So they just kind of talk about how you can use this to heal people from sound waves. So we'll kind of look at some of that healing stuff in a minute here. I just want to look at, just kind of recap what we talked about today. So DNA may be more wave-like than particle-like. We may be living in an energetic field that can store and transmit information. And last time we talked about, is this maybe a scalar field? David Wilcock in his book Source Field Investigations calls it the source field. I think modern science calls it the Higgs field. They will never admit that it's a scalar field or a source field. They're going to call it the Higgs field. DNA molecules appear to absorb light and electromagnetic radiation. Light and photons remain in place in this energy field for up to 30 days. That's known as our DNA phantom effect. And other indications of genetic information being stored or transmitted include the phantom leaf effect, embryonic programming, and pancreatic regeneration. So these are all examples where it seems that we can take an energy wave, encode it with healthy genetic information, and transmit that healthy information to a diseased or sick cell and heal the cell, or we can change it from a salamander to a frog. A diseased strawberry or raspberry plant into a healthy raspberry plant. A diseased pancreas into a healthy pancreas. All from information that's being carried by a wave. So this is just a quote from Dr. Garyaev. In particular, we have found it possible to regenerate endocrine glands in animals. By the same means, we have significantly curbed the aging process in human cells and even grown new adult human teeth in individuals who had lost them. Unquote. So, to me, that's just astonishing information. That's why I kind of want to maybe do a second part on this, but we'll see. I don't want to bore you guys completely. But I'm sorry, I'm just kind of going to look at this paper a little bit here. So the postulates of the new paradigm, or his new theory. All living organisms consist of two substances, the material substance and the energy informational or subtle substance. So I don't know if any of you are ever into like alternative science or healing arts. They'll often talk about a subtle body, and that subtle body is your energy field. So Dr. Garyaev here is postulating that same sort of thing, where we have the material substance or physical body, but we also have an energy body or an energy field around us, in us. We are both things. We have these two parts to us. The key property which distinguishes the energy substance and the corresponding energy field from all other substances and fields known in modern physics is that the energy substance is omnipresent, i.e., it is present simultaneously at each point in space of our three-dimensional material world. So again, that kind of sounds like our scalar waves that are everywhere all the time. You can communicate instantaneously because of these scalar waves. So there's some other postulates on here, but those were kind of the main ones that I really kind of stuck out, because we are kind of continuing this discussion of scalar energy and living in a scalar field and influencing this field with our own energy, and how we are energy. When you really come down to it, we are energy. We are all energy. And I just, like I said, mentioned before, how at conception there's light is produced. So when we're all born, as soon as we are created, there's a flash of light. I love it. So if you go back to Garyaev's site, we can look at, so here's the healing matrix. So I was going to mention this story about these healing waves. So a long time ago, and I have no idea where I heard this anymore, but there was a guy who had these sound files for different ailments, basically, and one of them was for weight loss, and I'm always struggling with weight. I don't know how long I can keep blaming my baby for it. At some point, I'm going to have to say that I just need to lose the weight, because right now I'm still like, well, I just had a baby, but I just had him 16 months ago. At some point, I'm going to lose the baby weight. So I've always been struggling, there's always been some sort of weight to lose. So this guy had a sound file that said if you played the sound. It would basically realign your body so that it naturally burned fat and calories like the way it's supposed to. And he had it for all sorts of things. He had it for joint pain. And the idea was that you play the sound file like through headphones, and you put the headphones on your joint, your knee, your ankle, your foot, and the sound waves would alleviate your pain. And he specifically mentioned to listen to it at a low enough volume that you can't hear it, because it is not pleasant to listen to, and it's not meant to be. It sounds like white noise and static, and it really grates on your nerves. Like, you can't stand to listen to it, and that was kind of the point. It was just that, and I don't really remember how he said it worked, but when I heard this healing matrix from Dr. Garyaev's website, I was like, that's the same sort of sound that I was listening to on that, the sound wave for weight loss, because it was terrible. You didn't want to listen to it, so you had to have it playing low enough that you couldn't really hear it because it was obnoxious. So I'm just going to play this. So this is one of his, it's a universal healing matrix, and again, it's in Russian. I just know that it says Garyaev's matrix. Let me just play a few seconds for you guys, and you can hear what it sounds like. Okay, so here we go. Okay, so that's just a few seconds there. But yes, that's a healing matrix. So I think this audio file you can find it on YouTube. I think it's like 30 minutes or something. But it's of just that kind of weird, staticky, white noise. Yeah, so I'll put this website link in the show notes. And again, you can, if you search for Garyaev Matrix on YouTube, and Garyaev, let me see if I can find his English spelling of it. So you can search for Garyaev, Garyaev. And again, I'll link his website. His website is wavegenetics.org. And you can find this matrix file on his website if you click on application. But yeah, so that's what the healing sounds like, his healing matrix. And he has a whole bunch of stuff on his website about having these sound files and producing a sound file from a picture, like of a baby picture of you, and you can basically extract the DNA information from when you were a baby, put it into a sound file, and then when you play it for yourself, that the baby DNA, where it's completely healed, there's no degradation, will then repair your aging DNA in your body. That's his theory. That's what he does. He's got tons of testimonies on here from people who have, I don't know if they've done that specific, but he's got other healing techniques and methods. He has an institute where people go to get treated, and there are really good reviews for it, but he's got tons of information. The site that we're on right now is called Information Medicine. So just to quote, treatment using information that is a quantum display of a healthy state of a particular person. So they're basically going to take a picture, a sound from a healthy DNA, play it, and then your DNA will become healthy. That information transmits to your cells. So it's pretty amazing. And I know I've kind of probably run too long on this episode, so I apologize if it's getting too boring for you guys. I hope you don't mind. I hope you are enjoying this. I love talking about this stuff. I love researching it. There's just so much to research, and so I feel like I'm going to end up doing 2-parters on everything. I'm going to try not to do that too much because I do want to cover other topics, but I love this stuff. So the next time I'll probably talk more about, I might do a little bit more about the linguistics aspect of this as well. We'll see. Like I said, I don't want to get to be too repetitive or boring. But I do love this idea of biofields, biophotons, the fact that we're all energy. I love all this energy stuff. I know it, but I just don't want to be boring to you guys. So let me know if I am. Again, you can contact me. I've got a Facebook group. You can join that. I'm actually on Instagram as The Interested Reader. If you want to find me on Instagram, I don't know how many people listening will use that, but I'm there. I've got a Facebook group. I've got contact forms on the website. You can get a hold of me kind of however you need to. Just send me questions, ask me about why I can't get a better setup for my lesson plan, but I'm working on that. So, I'll talk to you guys. Again, this episode I know is released late again. I can't seem to make the deadline. I don't know. Maybe someday. I keep setting the deadline, and I keep missing it every single time, so I should probably just change the deadline. So I'm just going to post this one. I'll just probably post it today. Today's Wednesday, March 6th. This was supposed to be out last Friday on the 1st. So I'll post it today. The next one is supposed to be next Friday on the 15th, but I guess we can just say don't count on that. Let's just plan on probably the 22nd. Yeah, it just takes me a while to get all my research done. I don't know. Anyway. Alright, so I hope you guys like this. I hope you're finding some sort of use from these podcasts. If nothing else, I hope you're just interested in something that, it sparks your imagination, or you want to question something and you want to go look into something, or ask me the question. I mean, if you don't want to go do the research, ask me to do it. I'll do it. I need topic material for the podcast anyway, so you may as well just make me do it. I'd love to. So yeah, let me know what you guys are thinking about. I hope you're enjoying this. Tell me if you're not. Tell me what else you want. If you want something different. I want to help you guys out. So let me know what you're thinking. And thanks for listening. Thanks for putting up with me. I love you guys, and I will talk to you at our next session. Bye, guys.
I hope you enjoyed this episode and are inspired to question and investigate the world around you. Let me know what you're thinking about. You can contact me through the website, theinterestedreader.com, send me an email at sarah@theinterestedreader.com, or join the Interested Reader group on Facebook. I'll see you at some later time T, where T is greater than T naught, and T naught is equal to now. Thanks for being an interested reader, and I'll see you next time.